And we are live. This is the TWIS Minion Science Island Hangout. And joining me are Dale. How you doing, Dale?
I am excellent.
Oh, sorry.
I am excellent, doing good. Look at my new magazine.
Ooh, Scientific America.
It's all good.
Gravity waves. We were trying to talk about that a couple of weeks ago. And we don't have, next week maybe, I don't know, Dale's going to be working on some music. Maybe we'll get some dancing music to start the show. I don't know. And from Finland we have Jani, representing Europe, our European representative. How you doing, Jani?
No. some magazine like this.
All right.
Anything Jani has is going to have something to do with computers. I know it. Virtual something or other.
He's the computer guy.
You know, I never introduced myself, and I probably should. Ed Dyer. Enough said. Let's get into the science stories. And the first one, we always have to start with a NASA. Space science seems to be our favorite topic. And here we're talking about NASA being able to simulate something called space dust. Not dust mites. This is space dust. And the idea and the real breakthrough is when you can recreate a phenomena in a lab on Earth, something that is occurring in space. In this case, we have this something called space dust, and this is occurring perhaps hundreds of thousands of light years away, and it's very, very hard for us to be able to observe. We're getting better at it, but it's hard for us to be able to observe something like space dust and what's happening in other planets and galaxies. But if we can create the same thing in a lab here on Earth, then we can study it better. And that's what they have done at NASA. They have successfully reproduced the process that occur in the atmosphere of a red giant star and lead to the formation of planet-forming interstellar dust. The dust grains that form around dying stars and are ejected into the interstellar medium lead after a life cycle spanning millions of years to the formation of planets and are the key component of the universe's evolution. And the scientists have found the materials that make up the building blocks of the universe are much more complicated than originally anticipated. Of course, the harsh conditions of space are extremely difficult to reproduce in the lab and have long hindered efforts to interpret and analyze observations from space. But now they've got something called the cosmic simulator. We can now discover clues to questions about the composition and evolution of the universe. Jumping ahead a little bit, the researchers can successfully simulate gas phase environments similar to interstellar stars, stellar envelopes, or planetary atmospheres environments by expanding gas using a cold jet spray of argon gas seeded with hydrocarbons that cools down the molecules to temperatures representing these elements. The chamber is the heart of the system. It recreates the extreme conditions that reign in space, where interstellar molecules and ions float in a vacuum at densities that are billions of Earth's atmosphere. Average temperatures can be less than minus 270 degrees Fahrenheit, about 100 degrees Kelvin. And the environment is bathed in ultraviolet and visible radiation emanating from nearby stars. By using this cosmic and building on the network, they recently published in Astrophysics, we can now for the first time truly create and visualize in the lab the formation of carbon grains in the envelope of stars and learn about their formation. The team they had started with small hydromolecules that it expanded in the cool jet spray in this cosmic and exposed to high energy in the electra discharge. They detected and characterized the large molecules that form in the gas phase from these precursor molecules with highly sensitive detectors. During cosmic experiments, we're able to form and detect nanoparticles on the order of 10 nanometer size, grains ranging from 10 to 500 nanometers, and aggregates of grains up to 1.5 micrometers in diameter, about a tenth of the width of a human hair. Amazing how the scale, the small scale they can work with. These results have important implications and ramifications not only for interstellar astrophysics but also for planetary science. For example, they could provide new clues on the types of grains present in dust around stars and help us understand the formation of planets. Very cool stuff. Amazing technology.
So they're creating dust in this cosmic chamber that simulates the conditions in outer space.
Yes.
Hmm.
I think, and the key to me is their ability to work at the small scales they can and with the extreme temperatures and environments that you find in outer space.
I wonder who would think of this. I picture somebody sitting around, a bunch of NASA scientists in a boardroom, and I've got an idea. Let's build this gigantic chamber, and we'll see if we can create space dust. That's awesome. So the space dust, they're saying, I wonder how the formation of planets work. Does the dust naturally sort of coagulate into larger and larger things as mass increases? I wonder if that's part of...
That's my guess.
Yeah.
It's kind of sounds like the space dust is kind of a fundamental molecules that are involved in the formation of planets and things. So actually, that's what we're going to find out, I guess.
Yes.
Yeah, when we're talking about the observations we make about distant objects, what we're really talking about, we're gathering light, correct? At this point, all we can do is gather light. Now, it's amazing what we can discern from just analyzing the light and things like gravitational lensing and
spectroscopy.
Yes. And you know, it seems to me we're making exponential advances. I mean, it's not these advances are not happening at a slow pace. They're happening at a pretty fast pace. The only thing blocking it is probably funding. It's not technology. It's not the ability of the engineers to think up these new technologies. It's funding.
You know, that's where worldwide, my understanding is a lot of the government-based organizations you know, that are funded to do this material science and the things that really aren't going to be that profitable.
Mm-hmm.
I see a lot of the universities and different, you know, governmental institutions around the world, you know, from my own sort of view, it seems like more and more they're trying to make them into profit-based institutions. But where, you know, I think you leave the profit-based, you know, making medicines and stuff, leave that to the private groups. And I think we need the government to do the things that aren't profitable to sort of spur on the technology. And we need to look at these things as an investment for the profitable groups to go ahead and make businesses out of these things. So it's kind of sad when you see them, you know, trying to force them to become like a profit center rather than trying to do real science and that sort of thing. So this is a good example of, you know, they're doing some real science. You're not going to find a profit-driven business creating space dust. So
yeah, and in the United States, very much a business-driven environment. What you mentioned, profits is two things can get you money for funding. Is it something that can make somebody a buck, or is it something the Defense Department can use to blow up people? That's the two keys. You got to get either defense. You get the Defense Department behind you, you got a blank check. Or we are seeing these entrepreneurs like Elon Musk, and is he behind SpaceX? Is that SpaceX? I forget.
I believe so, yeah.
And so you are seeing individual entrepreneurs who are using vision and investing in these technologies. But the real exotic stuff like this, there will, you know, you'll be able to make, there'll be something profitable coming out of it, but it might not happen for years. It might not happen for a century. You don't know. You just don't know.
Yeah, there's definitely a place for, you know, just raw research where you're not going to make a profit on it, but it does move us forward as a society. And there are, you know, the byproduct of this years of investment does create business and jobs eventually. And yeah, I think that's where the role of government should be, but I don't think they should be demanding that our you know, educational institutions and research centers should be kind of driven for just profit. That's for sure.
It's we're still, no matter the funding problems, we're still. I mean, every week, like this week, I still see just an amazing amount of science research happening. I'm going to jump to the next story, and this is kind of Star Trek, science fiction beginning to meet science fact. These are researchers who have built an acoustic tractor beam. Let me do the screen share. Now, this is a short article, fortunately, because I really don't understand what the heck is going on here. I will just start by going through. There's a nice little diagram. Incoming beams and propagation accesses, and I got no idea. So it says neoconservative pushing forces. Okay, Dale, you know what a neoconservative pushing force is? I got no clue.
A neoconservative.
Neoconservative pushing force exerted on an object by a plane wave, okay, as a result of strong backscattering. So I understand the words on a and object. That's about the only words I understand in that particular sentence. And two, decreasing the pushing force due to an enhanced forward scattering in a non-paraxial beam. The authors used a target designed to maximize the forward scattering of acoustic radiation, leading to a pulling neoconservative force towards the source. Now the article, the word neoconservative, I don't quite understand. But as this article talks a little bit further down, the problem is light, using light pushes things away. So they're, instead of using light, they're using acoustics as a way of doing that pulling action. In any event, tractor beams, as we all know, are a staple of science fiction. A beam is emitted from a spaceship that can be used to lock onto another object, such as another spaceship, and then used to move that other object in the direction. Okay, tractor beams seems counterintuitive, as beams of light tend to push objects away rather than attract them. However, prior research has shown optical tractor beams can be created in a nanoparticle level. They call them optical tweezers. In this new effort, the research team has extended the ability of tractor beam by using acoustics as opposed to optics. Sending a beam at an object and pulling it and having it pull the object closer, not just repeating what it said before. Now let's try this again. Sending a beam to an object and having it pull the object closer rather than push it can work because of the scattering of the wave that occurs when it collides with the object. And if the wave is sent at an angle to the object. So you're taking this wave, okay, you're taking the wave and you're scattering it. So it won't have that pushing force. If the scattering and the angle are controlled just right, a low pressure zone can be created in front of the object.
Oh, I see.
Now, pushing it back towards the origin of the beam. The researchers used ultrasonic sound waves in a tank of water. They put an array of ultrasound emitters at the bottom of the tank and used hollow isosceles. I don't know what this is. Isosceles triangle. I know that. Prism.
Yep.
The object is pulled. You know what they're talking about here? Dale, got an idea?
Yeah, I think I have an idea. So essentially, what's happening, I think, is if you put pressure from the outside angles. Yeah, that's a great diagram right there. So you can see the outside angles. It's approaching the target on the sides, and it creates low pressure in front. So if you considered it, you know, something is going to move where in the path of the least pressure, right? So if you have pressure coming and hitting it on the sides, there's going to be less pressure on the front, thus moving it forward. I think that's what they might be talking about. I'm interested in the optical tweezers because they based it on the same technology as that. So I just clicked on optical tweezers to see what maybe they did with that, and it really doesn't lead anywhere.
Oh, all right. Wikipedia, the source of all knowledge.
Yeah. So optical tweezers, yeah, I found it on Wikipedia, and it says it's are scientific instruments that use a highly focused laser beam to provide an attractive or repulsive force depending on the refractive index mismatch to physically hold and move microscopic dielectric objects. So dielectric is generally something that doesn't conduct very well. So the refractive index, depending on it, and I think they're just talking about there's the angle of incidence, but it's being refracted through something, creates that mismatch and then creates that low pressure in front of the object, and it essentially moves towards you because you're adding pressure to the sides and rear of it. So they use refraction, and if you remember refraction from shining, I believe like if you shine light through water, it refracts, right?
Right.
And I think they're using that refraction to create more angle, so you're getting almost, you know, as much to the rear, creating as much pressure to the rear and to the sides that it basically wants to move forward because there's nothing there. I think.
That's interesting. Good explanation. I can visualize. I can definitely visualize.
Well, I hope someone will complain and say we got it all wrong, and then they can come on the show.
Jani has an article about Mars gardens. I think we've talked about this, building Mars gardens, veggie gardens in space, and how we're going to space veggies. I think well, they're building, they are growing vegetables, correct? On the International Space Station, I think. So we can study the effects of anything growing, even spiders creating webs, and then learn about it from the space station.
See, now NASA, what NASA could do here, right? If they're thinking, they're always looking for funding, and this is a little bit of a joke here, but okay, so something, the way it gets value, right, is it becomes something rare is very valuable, right? So grow space veggies in outer space, bring them back to Earth and sell them, right? Because there's very few of them. You could sell them like $100,000 for a piece of broccoli, right? And that would fund.
I'm sorry, I'm interrupting you. We may have an expert. We have Amy Lynn joining us. Hopefully, right. She tried to join in the question and answer session. Didn't work. Amy, can you turn off your mic? Can we hear you speaking? Hello, Amy. We're not at the right angle, perhaps.
We need to create an area of low pressure through
Yes. Can you see this? I'm looking in the question and answer session. It says, can I see this? Well, yes, I can see that. So I guess Amy wants to post this. I should say that a new message in the chat area. Okay. Communications. And she's saying it's non-conservative, not neoconservative. I got politics on the brain. There we go. There's something else going on in the background so the chat works. Okay. Well, we will pay attention to the question and answer, the little Q&A button, and maybe at some point you can join us, if not live. Well, you can join us live. Maybe you can do it acoustically. We're doing it optically, and perhaps you can join us acoustically.
Yeah, it shows that her mic is muted, but she's on, right?
Yes. She's here. Okay. Communications. Hello, hello. This is an experiment. I love this. This is an experiment. We're experimenting with stuff.
Yeah. Like space veggies.
Okay. Let's see. Okay, Amy, if you can get to the chat area, I guess my question would be, can you join us by voice, or would you just prefer to do it through question and answers? In any event, moving on to the next story. And Amy, as I say, was the one who sent that story to me about the tractor beams.
Cool.
Amy, very interested in space. She's part of the Space Buddy team with A.J. Smith, I think Ulysses. I'm very definitely interested in. She sent us stories before, so thank you, Amy.
Well, I wish she was on, because I want to know if we were right about this low pressure thing.
That would be cool.
All right. This next story was one, Dale, that you sent me. And it's an interesting. Two-lock box delivers cancer therapy. And this is from Rice University. And they have designed a tunable virus that works like a safety deposit box. 2 keys to open it and release its therapeutic cargo. Now, they've developed an adenoid-associated virus that locks only in the presence of 2 selected proteases. Proteases enzyme that cut up other proteins for disposal, because certain proteases are elevated at tumor sites. The virus can be designed to target and destroy cancer cells.
Cool.
There you go. Our friend the virus. Now it says these AVVs are fairly benign. What does fairly mean? It had become the object of intense study. And the researchers often try to target these AAVs to cellular receptors that might be slightly overexpressed in diseased cells. These guys at Rice took a different approach. They were looking for biomarkers beyond cellular receptors present at disease sites. In breast cancer, for instance, it's known that tumor cells over-secrete extracellular proteases. But perhaps more important are the infiltrating immune cells that migrate into the tumor microenvironment and that start pumping out a whole bunch of proteases as well. That's your delivery system. Well, that's what they're talking about. This is, I can't pronounce her name, Zhonghee Su. Their lab genetically inserted peptides into the self-assembling AAVSs to lock the capsids, the hard shells that protect genes contained within. The target proteasis recognizes the peptides, chews off the lock, effectively unlocking the virus and allowing it to bind to the diseased cell. If we were just looking for one proteasis, it might be at the cancer site. It could also be somewhere else in your body where you have inflammation. This could lead to undesirable side effects. By requiring 2 different proteases, let's say proteasis A and proteasis B, to open the locked virus, we may achieve higher delivery specificity, since the chance of having 2 prostheses elevated at the site becomes smaller. In future, molecular imaging approaches will be used to detect both the identity and concentration of these elevated proteases. With that information, we'll be able to pick a virus device from a panel of engineered variants that has the right properties to target the disease site. Sue said elevated proteases are found in many different tissues. She suggested these proteasis-activated viruses may be useful for treatment of not only cancers but other neurological diseases such as stroke, Parkinson's, and Alzheimer's disease. That's interesting. Using the viruses as a delivery system.
That is cool. So, so what's happening is, so a protease, when there's certain proteases that are expressed in cancer cells, and when this virus, and virus is like a strand of RNA with some instruction code, and it's carried in an adenovirus, which is, they use that to basically get into the cell.
Mm-hmm.
It's like a transport truck that'll allow it in the cell. So many times for gene therapy, it's really tough to actually get the payload of whatever code you're sending in the cell. So they use this kind of harmless virus that has very little in the way of negative pathological effects. And it gets into the cell, it sees these particular peptides, and in its presence, then this structure can release its payload. And that cargo could probably be like chemotherapy drugs or whatever they might want to deliver. So I guess they can target this with different types of things, because if the peptides that it's bumping into, now the proteases must chew at this thing and basically unlock it. Because an enzyme is basically, it eats stuff up, right?
Mm-hmm.
And so it must eat away at our little cargo truck here, this adeno-associated virus, and then that's how the payload is opened up. And I love the little diagram. They have a little Pac-Man on it, and I think that's representing the protease. And I believe you can always tell an enzyme because it has that ASE on it, right? So you can tell that, yeah, that's going to eat up something and have a reaction. So that's really cool if they can figure that out. Now, did they, as you were reading, did they do this? Where did they do their experiments? They were in a petri dish? Did they mention?
Different approach, biomarkers, capsids.
I never get too excited about these things, unless it, you know, at least if it was done in a mouse, they're getting a little closer, right?
I don't see. Let's see, the work appears this week. I'll click on that link. There's the abstract. I don't know if I can get to the full text. Let's see. Let me screen share. There's the abstract. Supporting info. Can I get to the supporting info? Oops, it's a PFD.
So that it's someone had a sense of humor, because that diagram is, they show that they represent the proteases with a little looks like Pac-Man eating away at the capsid of the virus vector. So someone had a sense of humor.
Yeah, it doesn't. It's, you know, I have to purchase to get the full text. So
now I've seen this done before. I don't know. You know the paper folding. What's that called again?
origami.
Origami. Yeah. So there's something called DNA origami developed quite a few years ago, and they used DNA to engineer shapes. And I remember one of the first shapes they made were these tiny little happy faces made from basically using the rules and functions of DNA to create these shapes. And I know one of the things they accomplished years later was making something that could carry a payload like this.
Mhm.
And so I've seen this before. I wonder if it's sort of, if they're using that same sort of technology, because basically it came down to software programming to create the AC's, G's, and T's in a certain order, so that you would have a structure fold in on itself and create this sort of cargo transport for therapeutics. So that's interesting.
An interesting thing is that how they can get this virus payload. It's completely benign or fairly benign until it hits the specific. In fact, it's seeking out the particular cancer cells based on these proteases. Proteases. A particular kind of protease, I guess, produced by different kinds of cancers. I'm guessing they're different kinds. And so it's a way of you're really seeking out, you're specifically targeting, you're looking for and finding the cancer cell, and you're not delivering that package, that treatment, until you find the cancer cell. And it's the enzymes around the cancer cell or in the cancer cell that are actually opening up the packet.
Yeah, and the enzymes are the proteases kind of munching on when the package gets there, they munch on it inside the cell, and they get inside the cell because it's a virus. That's what a virus does. That's your entryway. There's only a couple ways in genetic engineering that they have to get these things in the cells, and the one I know is a virus. So they reprogram a virus to do what they want to do. So they use a very calm, mild thing that doesn't hurt us to get into the cells. And another way, they use high pressure, like it's just blasting the instructions basically, and doesn't sound very pleasant.
It doesn't sound like that. This particular process, it sounds to me like it would be then have little or no side effects.
I think there'd be, my guess, would there be some, because there's good, they overexpress these proteases in these cells, but it probably won't be perfect, right? Because there's even in normal cells, there's gonna be some of these proteases and the same peptides are going to exist.
So I make a mistake. The virus may target a healthy cell instead of a cancer cell.
But I bet you the ratio, like when they give chemotherapy to people, I believe the way it works is they're attacking all the fast dividing cells, because cancer quickly divides, right? So chemotherapy goes after the fast dividing cells, and they're targeted that way. That's why your hair falls out, because your hair has fast dividing cells, right? And that's why your hair falls out. Or you get a stomachache, because the lining of the gut, again, is another area with fast dividing cells, so chemotherapy tends to hit those areas hard. I think in this case, the great thing is the ratio would probably be very low. I bet you it would hit mostly cancer cells, and you'd get a little bit sick, but probably not very much, I bet, because it's going to go after the ones where it's going to be attracted to those. If it hits some of these areas and it doesn't bump into enough of these proteases, it won't unlock the cargo. But it's more likely to unlock the cargo where you have these Pac-Men munching away at the locks of this cargo transport.
Well, I wonder if there might be different kinds of proteases for different kinds of cancers.
Yeah, that's exactly what they said, I think. So
and if you can find the right profile, I mean, here you're getting very specific. You're using, your treatment is going to be very specific. You're dealing only with certain specific cells. That was the whole deal with targeted cancer treatments. This sounds like it's certainly preliminary, so I wouldn't expect this to be. I mean, even if it does work out, I get. Well, you'd have to start with mice. It doesn't say if they've started this treatment at all with mice and then primates and then people. I wonder, could you go just? I don't think you could just jump to people, could you?
Well, I know there's been scientists in the past that experiment on themselves, and you know, they believe it's littered. Science is littered with that, right? They believe so strongly in what they're doing that they'll begin to experiment on themselves. A lot of ethics there, a lot of probably legal issues. But you know, more and more, I think the landscape of testing, I think because of our knowledge, it's going to get more and more reliable. That short circuits and shorter paths to getting things approved, because we're getting a lot more. The map is a lot clearer now than it's ever been. So
yes.
It's getting very predictable, and a lot of times now they use the word engineering all the time, and it's because that's the way they're looking at it. The molecular pathways, they're beginning to understand what will happen, and they can simulate it, you know, beforehand. And I think that's the world we're getting into, where, you know, the time it takes to get a drug to market, or genetically reprogramming cells. They just did it with the liver in, I think it was a monkey, or I can't remember what. Do you remember what it was? I think we talked about it.
Let's see.
They reprogrammed it, basically.
There was the mice study, and then there was the monkey study. And I'm trying to think, the monkey study, can't remember.
That's a tough one, but they did reprogram
the liver
and essentially cured the disease by changing the nucleotides around. So I think that's where we're headed to, and that is so exciting, you know, to be able to just reprogram our way out of it with very little, you know, pathological effects. Yeah,
we're beyond saying, okay, we got this thing, let's try it on somebody and see what happens. When you're developing it, you have so much precision and so much knowledge beforehand at the development stage. When you finally reach the point where you think you may have the final product, it's really, really been fine-tuned to a much higher degree than in the past. And that should shrink that time frame, I think, between development and actual application.
And that's why I always mention this CRISPR-Cas9 technology they've just come up with. It's so exciting because we've been able to read DNA for a long period of time, but it was so hard to write it. It was so expensive and inaccurate. And this new technique, they can rewrite tons of these nucleotides very, very simply. And so when you can reprogram something and then examine it and watch what happens, that's exciting, because now they can get a better understanding by just simply, let's rewrite the software and see what happens to this bacteria or whatever you're dealing with. And we've never been able to do that. It was tough. They would use zinc finger nucleotides, which was very difficult. You need highly trained specialists to do this. And now we're in this new world where our internal hard drive can be rewritten much simpler than ever before and much more accurately. So I'm excited.
Yes, that's right. Dale and Jani, everything is just software.
It is. Everything's information. We're in the matrix.
I'll move on to the next story, talking about scientists creating, come on, let me get there we go. Scientists creating the first living organism that transmits added letters in DNA alphabet, and this is the Scripps Research Institute. Let's see, they have engineered a bacterium whose genetic material includes an added pair of DNA letters or bases found in nature. The cells of these unique bacterium can replicate the unnatural DNA bases more or less normally. All right. Does that mean these bacterium do not exist in nature?
Yeah, that's exactly what that means.
Okay. I don't want to be okay. Life on Earth and all
I guess they do now.
In fact, actually, if you don't mind, Dale, maybe you can go over the story.
Sure. You got the first paragraph done, right?
Yes.
Okay, so life on Earth in all its diversity is encoded by only 2 pairs of DNA bases. So you have A binding to T and you have C binding to G. And what we've made is an organism that stably contains those two plus a third. So they've put an unnatural pair of these bases together that they've synthesized. This shows that other solutions to storing information are possible, and of course, it takes us closer to an expanded DNA biology that will have many exciting applications, from new medicines to new kinds of nanotechnology. The report on the achievement appears May 7th, 2014 in an advanced online publication at the journal Nature. So that's a pretty good journal.
So they're talking about re-engineering. This is all re-engineering bacteria.
It is, and I know that's one of the things, I think Craig Venter, George Church, like all those characters that are sort of at the leading edge of this, that was kind of the dream, was can we create two nucleotides, a paired base that would extend our capability so that all sorts of new possibilities would occur. Because
that's amazing. We're changing DNA itself?
Yeah, that's exactly it.
Wow. So
they've created their own chemical structure, and the key there. So you have DNA, which the central sort of thing within in biology is all of in genetics is DNA becomes RNA. That RNA gets fed like a ticker tape through something called a ribosome, and that spits out peptides, which create a protein. And proteins do everything in our body. Yeah. So if you can imagine now that they've created this new synthetic pair of letters, that means they're going to create new types of protein structures that will do different things, and it opens up the whole world to a whole bunch of new interesting things, and it's very interesting. So they said there's many challenges. Rosenberg and his laboratory have been working since the late 1990s to find pairs of molecules that could serve as new functional DNA bases. So they've been working at this for a while. So they said, in principle, it could code for proteins and organisms that have never existed before. Yeah, just like I was saying. New proteins that have never existed, they're basically creating something that hasn't been there before. The task hasn't been a simple one. Any functional new pair of DNA bases would have to bind with an affinity comparable to that natural nucleoside base pairs, adenine, thymine, and cytosine and guanine. Such new bases also would have to line up stably alongside the natural basis in a zipper-like stretch of DNA. So that sugar and phosphate backbone, they would have to be able to bind and coexist with the natural bases that are there. So they'd be required to zip, unzip, and rezip smoothly when worked on by natural polymerase enzymes during DNA replication and transcription into RNA. And somehow these nucleoside interlopers would have to avoid being attacked and removed by natural DNA repair mechanisms. I forgot about that, but
I like that word, somehow.
I know, and it sounds like they've done it. So we have all these DNA repair mechanisms that are looking for these foreign entities and bodies. So they've finally come up with something that's kind of worked around that. So it won't get repaired out while they're trying to create these new proteins.
I can see two reactions to reading this. The first is, wow, how cool, what an amazing set of possibilities, and the others being an absolute horror, Frankenstein. You know?
It is. Well, you know what? We're down that path, no matter how you look at it. And this is just
that's true. That is true.
And in some ways it concerns me, in other ways it's exciting.
That's always been true about medical research, anyway. There's always been, a good side and a bad side to it. But here, man, you're creating something new in DNA itself.
Well, here's the kicker. They said a little bit further down, it said these unnatural base pairs, it work beautifully in vitro. So you're in a petri dish, right? And the big challenge is getting it to work in a living cell. So it's interesting they've done it in a petri dish, and boy, what an accomplishment. But will it survive a living cell with all the other mechanics in there? I don't know. So it was done with E. coli. They inserted it into E. coli, which is a little factory all into its own. the plasmid DNA contain the natural TA and CG base pairs. So those letters are like magnets. T always goes with A, and cytosine always goes with guanine. And they put it along with those base pairs, and they put the unnatural base pair in there, the newly synthesized one. Why do they give it these complex names, though? These people need to learn about promotion. Take away the complex names and give it some new names like Sparky and Frank or something.
The last article in NASA, they were talking about cosmic. You know, they used an acronym. They probably made the acronym up so it would spell cosmic.
And you know what? Cosmic is awesome. But they need to almost take a page out of the NASA promotions department and give it something cool. And I'm not kidding, you know, to get people interested. I'm sure these are very appropriate names if you have a PhD.
It's really hard to appreciate what they've actually done here in creating a completely new type of bacterium.
Oh, yeah, it's very impressive. Obstacles all over the place. But yeah, that's about it. I kind of just glanced through the rest, but I think we hit the nail on the head. The only thing we didn't mention was because I was talking about DNA origami or origami, however you pronounce it, the paper folding. This method of DNA and creating nanotechnology using DNA, this would open up, there'd be applications there too, because if you can synthesize it with a new base pair, that means you're going to open the door to creating new structures and different types of nanotechnology. So that's very important for that too.
Yeah, and they got the E. coli, they got it to reproduce. It was able to reproduce. So you've got this bacterium that is able to reproduce itself.
Yeah, that's crazy. That's crazy neat.
They say that at the end, the next step will be to demonstrate the in-cell transcription of this new expanded alphabet DNA into the RNA that feeds the protein-making machinery of cell. Okay, now they said they've got the bacterium, this new bacterium, to reproduce itself. E. coli. They've got this E. coli can reproduce itself. Doesn't that mean that they've got the RNA process involved?
Well, it kind of must be my sketchy knowledge on some of this, because it's almost what they do and they don't. I don't quite understand. So they must be, maybe they've done this, in vivo as well, and there's a difference where they're talking about two different things there, or it's just my crazy misunderstanding of these very, very smart people.
Hello, AJ. Are you there? He's unmuting himself. Now, see, I see two people wearing T-shirts.
All right.
3 people wearing T-shirts. I'm the only one not. I'm the old man who's probably too cold, and I got to wear the shirt. How you doing, AJ?
I'm doing great. How are you doing, Ed?
That's good. How'd your car make out?
Which one? I got it here.
Oh, that's great.
So that was a big step in the engine swap.
That's good. We got an engineer here.
Engineer would help with engines.
I'm surrounded by people who are in the programming mode. All you guys are in the programming mode.
I'm actually making a program right now, too.
You're making a program now as we speak. How cool.
What are we talking about?
Well, we were just talking about creating a new kind of living organism, a new kind of bacterium. The next story, this is, it's a spurt of heart muscle cell division seen in mice well before birth, and the implication for repair of congenital heart disease. Let me screen share this. That didn't work. Come on, I am not the programmer here. There we go. There's our friend Science Daily, and this is from Emory Health Institute. And the entire heart muscle in young children may be capable of regeneration. In young mice 15 years old, cardiac muscle cells undergo a precisely timed spurt of cell division lasting around a day. This previously unobserved phenomena contradicts the long-held belief that cardiac muscle cells do not divide after the first few days of life. Learning how our reproductive system works. And the thing here is, if we can learn precisely how different organs in the body, when they're developed, learning more about how they're developed, then we can repair them and create them. These researchers at Emory University discovered that in young mice, cardiac muscle cells undergo a precisely timed spurt. The total number of cardiac muscle cells increases by about 40% during this time, when the rest of the body is growing rapidly. And a 15-day-old mice is the equivalent of a child in kindergarten. The burst of cell division is driven by a surge of thyroid hormone. This suggests that thyroid hormone can aid in the treatment of children with congenital heart defects. In fact, the doctors have already tested thyroid hormone supplementation in this setting on a small scale. Broader hints developing therapies for the heart. Activating the regeneration potential of muscle cells themselves is a strategy that is an alternative to focusing on heart's stem cells. Let's see. The researchers tested how mice at the age of day 15 can recover from blockage of coronary artery. Consistent with previous research, newborn day 2 mice showed a high level of repair after such an injury, but at day 21 they did not. The day 15 mice recovered more than the day 21 mice, indicating that the same repair is still possible at day 15. The discovery came unexpectedly during the course of their investigating the role of gene c-kit. Dale, ever heard of that?
Gene, say that again?
Gene C-KIT, c-kit, an important marker for stem cells. Never heard of it before.
Never heard of it.
For stem cells in cardiac muscle growth. Adult mice with a disabled c-kit gene in the heart have more cardiac muscle cells. They want to know why the difference appeared. We started counting the cardiomyocyte cell numbers. Excuse me?
Cardiomyocytes, is that?
Cardiomyocyte, thank you. Cell numbers from birth until puberty. And it was interesting to see the numbers increasing so sharply in just one day. It turns out that this c-kit deficiency and wild-type mice both have a spurt of proliferation. Differences between them appear later. Previously, investigators did not see this burst of growth because they were not looking for it. Even if in humans the proliferation of cardiac muscle cells does not take place in such a tight time frame as it does in mice, the finding is still relevant to human medicine. So learning how, in this case, heart muscles, how they grow, when they grow, that is a key for me. Actually, I want to know brain muscles, brain cells. I wonder when brain cells start to develop. More precisely, how brain cells start to develop.
Half of mine never developed properly.
Mine developed properly. They just died off too quick. Just dying off.
So it seems like what they've discovered is undifferentiated stem cells are still active at an early age. So if you have defects, those undifferentiated cells can differentiate into cardiomyocytes, which is if you look at cardiomyocytes, they beat, right? The heart cells. And that's interesting. Now I know they've done stem cell injections into the heart, and they've had some really interesting results in I think they're phase 2 trials, where they've actually taken, traditionally it was kind of once you have some scar tissue in your heart, that's it. You're not going to fix that. But they're having, what they're seeing is that, I don't know if that has to do with this, but they're finding that you can inject the stem cells and get it beating again. So that's interesting. So by learning this, the c-KIT gene, I was just looking that up, and so it's a protein in humans encoded by the KIT gene. And the cell surface marker, I believe. I'm just kind of glancing through this really quickly.
My question is, what is the trigger that causes that undifferentiated stem cell to actually start growing into, start dividing into heart, muscle, liver, whatever? What's that trigger?
Well, I guess that when you're born and you have cell division, after conception,
does that cell division happen on a regular? It's a very regular schedule that these cells develop into different organs, specifically at different times. They're talking about day 15 as opposed to day 21.
Isn't that interesting? That's very interesting. They're definitely onto something here, and let's hope they get funded. Who is it that's doing this research? Emory Health Sciences. Or no, was that the publication or something?
Oh, it's Emory. Emory is the name of the university.
Okay.
Where this research is going on.
And they got published by Cell, so that's impressive.
Mm-hmm.
Yeah, no, stem cells, boy, if we can become masters at inducing them back to a pluripotent state, that would be ideal.
Yeah.
Mm-hmm.
That would be
Yeah,
great.
that's taking that was the primate study I talked about last week, and I forget what organ we're talking about, where they're able to take the adult stem cell from your own or from the body of a primate and use that to turn it back into a pluripotent stem cell.
Yeah, that'd be wonderful, and for so many different reasons.
Mm-hmm.
And I know they found out, I believe, one of the keys, like after differentiation occurs, is the extracellular matrix. That's sort of a template. The structure of it is really critical, and that's really interesting too. None of this is simple, but we're rapidly learning. Mostly because of programmers like AJ. They're solving all the universal problems for us because it's all computational now.
I don't think apps are going to solve anything.
Hey, we are apps. Our genes are the apps.
Sure. World is a program.
What aperture science?
Yes, it is. Alright. Okay, now we're going to wander off into the environment. And it's too bad our friend Ulysses isn't here, because I know he would have fun with this kind of a story. And this is from the University of Illinois at Urbana. What they've found is they looked at multiple varieties of wheat, rice, field peas. I didn't know there were field peas. Field peas, wild peas, as opposed to peas that grow on a vine. Soybeans, maize, sorghum, grown in fields with atmospheric carbon dioxide levels like those expected in the middle of the century. Atmospheric CO2 concentrations are currently approaching 400 parts per million and are expected to rise to 550 parts per million. It's interesting how they were able to create this experiment. They simulated CO2 levels in open-air fields using a system called the Free Air Concentration Enrichment. There you go, a nice little acronym called FACE. That's what you do. First you create the word you want them to have it, and then you create the acronym.
Yeah.
Which pumps out, monitors, and adjusts ground level atmospheric CO2 to simulate future conditions. In this study, all other growing conditions, sunlight, soil, water, and temperature, are the same for plants at high CO2 and those used in the control. Experiments reveal that the nutritional quality of a number of the world's most important crops dropped in response to high CO2 levels. They say the study contributed more than tenfold more data regarding both zinc and iron content in edible plants. And this was a study that was performed in many different countries around the world, not just in one place. And the zinc levels went down significantly in wheat, rice, field peas, and soybeans, but wheat and rice also saw notable declines in protein content at higher CO2. And the levels we're talking about, I think, are something like between 9 and 5% decrease in zinc and iron nutrients. Across a diverse set of environments in a number of countries, we saw this decrease in qualities. Nutrients in sorghum and maize remained relatively stable at higher CO2 levels because these crops use a type of photosynthesis called C4, which already concentrates carbon dioxide in their leaves. No idea what that means. C4 is a sort of fuel-injected photosynthesis that maize, sorghum, and millet use. Our previous work at Illinois has shown that their photosynthesis rates are not stimulated by being elevated at higher CO2. And here is a little bit more details on the study. Elevated CO2 levels affected different crops in different ways. Zinc, iron, and protein concentrations in wheat grown at high CO2 levels fell by 9.3%, 5.1%, and 6.3%. Field peas and soybeans also lost zinc and iron as CO2 levels rose. So we're talking about how much this decrease in nutrition is. At this point, from what I can see, you're talking around 5% to perhaps 10%. we know the climate's going to change. We're now coming up with experiments like this so we can see what the effect will be in different environments. In this particular case, they're working with higher levels of CO2. I got to wonder if that spike, and I think for like 600,000 years, CO2 levels never got above like 2.8%, maybe 3.8%. And then all of a sudden, the last 50 or 60 years, it spiked to that 4 level. It's continuing to go up. Obvious indication that something happening in the last 50 or 60 years. That's something most likely that's adding CO2 is us, stuff we're doing. Not good news unless we start. Well, we can do something. We can certainly do some things to reduce the amount of CO2 we emit into the atmosphere. How quickly that can slow down this rise in CO2 levels, that's kind of the key. And I don't really see any signs that we're going to be doing anything significant to change the amount of CO2 we emit into the atmosphere by the way we produce energy. So I would expect their estimates could well be right. Maybe CO2 levels will jump up to 550 parts per million. All right. Now, after that exciting story talking about decreasing nutrient levels, I know something we can talk about, real positive. How about the Black Death? Why do we talk about the Black Death?
So whoever came up with that word, they have a great promotions department. It's very simple, very, very effective.
And this is new research. It's what they're discovered is we could pretty much guess at. It's I guess the way they were able to fine-tune it, and they were looking at, this is the University of South Carolina, reported in Science Digest. They analyzed data, genetic analysis of 14th century Y. pestis, which is the plague that created what's called the Black Death, has not revealed significant functional differences in the ancient and modern strains. This suggests that we need to consider other factors such as the characteristics of humans in order to understand changes in the disease over time. To better understand these human factors, DeWitt spent the last decade examining skeletal remains of more than 1,000 men, women, and children who lived during, before, during, and after the Black Death. The skeletons are maintained in the archives of the Museum of London. The skeletons were cataloged in 3 feet by 1 foot boxes. As she studied each skeleton, she determined the biological sex, age at death, and analyzes specific markers, including porous lesions and teeth, to gauge each individual's general health. Her bioarchaeological, I can't say it.
Wow.
There we go, guys. You know what I mean. And they're coming up with these new words. They should, instead of coming up with longer words, they should come up with shorter words. You know, biology and archaeology and I don't know, whatever. providing a new dimension to the study of the Black Death and provides the first look at the lives of women and children during this medieval period. It's an innovative because of the analytical approach I took. My Black Death research is rare because the samples I used are exceedingly rare. There are only a handful of large cemetery samples that are clearly linked to the Black Death, and most of the prior record only deals with the experiences of men. So that was an important part of her research. Here's what she came across. The 14th century Black Death was not an indiscriminate killer, but instead targeted frail people of all ages. Okay? Survivors of the Black Death experienced improvements in health and longevity, with people living to ages of 70 or 80 years as compared to pre-Black Death populations. Improvements in survival post-Black Death didn't necessarily equate to good health over a lifespan, but revealed a hardiness to endure disease, included repeated bouts of plague. So you have that principle, whatever doesn't kill you makes you stronger, I guess is what we're talking about. It's interesting. So that the survivors, it wasn't that the survivors could resist illness necessarily more, it's that they could endure these illnesses more. In Black Death, either directly or indirectly, very powerfully shaped mortality patterns for generations after the epidemic ended. She was surprised to see at how much of a change she estimated between the pre- and post-Black Death periods. The Black Death was just the first outbreak of medieval plague. Despite this, I found substantial improvements in the demographics and health following the Black Death.
So were they saying that it helped out mankind as a whole? Like, is the hardiness to the resistance to the diseases passed down to their children?
Yeah, the one is the ability to resist disease, and there had to be some kind of immunity involved.
Right.
I would think. But what she is talking about is that the mortality rates, the increase in mortality rates, the living longer, had more to do with the ability to survive, a hardiness to survive illness. As opposed, for instance, these people had poor teeth. They had other signs of poor health.
Yeah, I'm just wondering if that would be passed down to your children.
Well, survivors are the ones that are going to spread their gene, so yes. Alright. We're getting close to the 10:25 mark. I want to wrap it up a little bit. I threw in this next study because it was mind-boggling about the U.S. healthcare system. And it's something me and Dale talked about in the pre-show, how the United States healthcare system really not working so well compared to Canada and Jani's Finland. This was reported in Atlantic magazine. Now, basically, I mean, the map, if you can see it, right off the bat, it is dramatic in terms of the quality of overall healthcare. And this is overall healthcare. Now, it's reported in something called the Commonwealth Fund. I don't know what kind of a bias, if any, this Commonwealth Health Fund has. But it looks like It was a fairly extensive study. I mean, right there in the South, all southern states would have the poorest health care.
Yep.
And little old Nevada. It's kind of weird. I don't know why Nevada would have such a poor health care system. But all these southern states. Texas, that's a surprise. I can understand, okay, New England. New England is small. Okay, Connecticut, small state. These are small states not dealing with large populations. Minnesota, the Midwest, Wisconsin, South Dakota. South Dakota, but not North Dakota. Well, South Dakota's system being better than North Dakota. But these are smaller states. California, pretty good healthcare system, a large state. New York, a large state. But Texas, why the heck is Texas's healthcare system so poor? Why are the people of Texas apparently in poorer health overall than people in California? I don't know.
I have a question for you.
Yes?
I don't know a lot about population in the U.S., but if you go back to that map there, so the white areas is where they have better performance, right?
Mm-hmm.
So are those sparsely populated areas compared to the really dark ones? Like, is there a trend where population is having an influence on performance? Or is that not?
Well, you're right that most of those states with the highest level of health care are lower population states. However, you look at California, CA, that's a very highly populated state. New York, very highly populated state. And you compare that to Texas, which is very poor, and Texas is a very highly populated state. So you have three of the highest states in terms of populations. New York having the best, California having that third tier, and Texas having the fourth tier. And all of those states, you know, their populations are high, so it's not a simple case of high population. Nevada has a low population. Now, maybe there's something strange going on in Nevada.
This kind of made me look up something real quick because it reminded me of an older map, and if you'll keep that one up, I'll screen share mine.
Okay.
Does it have any correlation?
Well, the same states that are red are also black in the first map.
But this was, what, back in 1860 when there was a division between the Confederates. It's the same map.
Some similarities. The only thing is, you talk about the Confederate States, I mean, you're talking about Civil War. So,
I know, and they're still...
Amy is saying that, no surprise that Florida is one of the poorest, also has one of the worst school systems. And the thing that...
I'm just kind of wondering if a lot of times the larger an organization gets, the more red tape that occurs, the more inefficient it becomes, with exceptions. I think there's some large organizations that manage to pull it off, but I was just wondering if there's a correlation between that. With a larger state, you have larger government institutions managing things, and if there's a correlation with that.
My guess would be, because I see differences between California and New York and Texas. Florida's got a fairly good-sized population. What was Pennsylvania? That's got a fairly good-sized population.
Texas too.
So Texas and Florida. Florida probably has a lot of elderly people, but so does Arizona. And Arizona apparently has a pretty good healthcare system, as or the people in Arizona appear to be in perhaps better health. Another aspect of this, the one I find most disturbing, is that racial and ethnic minorities in Arkansas, Georgia, Indiana, Mississippi, and North Carolina face some of the widest disparities relative to the national average across all of the indicators. Mississippi is already at a disadvantage. A black man in Mississippi has a shorter life expectancy than the average American did in 1960. The obesity rate is 35%. Now, it has high poverty, and certainly there's going to be a correlation between, I'm thinking, high poverty and health care.
That and fat's cheaper than protein.
Yeah. Where this could become relevant, where it is that in these states where you have poor health care, I'm generalizing, these states tend to be places where they don't like government programs. They would tend to oppose government health care programs. Now, Arizona is the same. Arizona is a conservative state, and they've got they're apparently got a pretty good health care system. So it's not an absolute, but it seems to me in general with these southern states, mostly all southern states, it looks like to me, these are all states that oppose health care. We have the recent, we have the health care, the affordable health insurance system that Obama's trying to implement. Off to a rocky start. Hopefully things getting better. But these southern states all oppose it, if I'm thinking. Maybe not all of them, but most of them don't want to take part in that lower affordable health care insurance. So that tells me this is likely to get worse before it gets better.
Could be another civil war.
It started here, folks.
I don't know. It's very disturbing. This probably was one of the most. I'm an optimist, but that was one of the most disturbing articles I read in quite a while. I guess we can cover. We'll go to something interesting in terms of getting more power out of solar cells. And this might be the last story we cover. Unless somebody else has something they want to talk about. And this is from our friends at MIT, getting more electricity out of solar cells. The new MIT model can guide the design of solar cells that produce less waste heat and more useful energy. Sunlight shines on today's solar cells. Much of the incoming energy is given off as waste heat rather than electric current. In a few materials, however, extra energy produces extra electrons, behavior that could significantly increase solar cell efficiency. The MIT team has identified a mechanism by which this phenomena happens, yielding a new design guidelines for using these special materials to make high-efficiency solar cells. And this was reported in Nature Chemistry. In most photovoltaic materials, a photon delivers energy that excites a molecule, causing it to release one electron. But when high-energy photons provide more than enough energy, the molecule still releases just one electron. Plus, all the other is waste heat. A few organic molecules don't follow that rule. Instead, they generate more than one electron per high energy photon. This is called singlet excitation fission. It was first identified in the 60s. However, achieving it on a functional level has proved difficult. Over the past 4 years, Van Voris and Baldwin have been pooling their theoretical and experimental expertise. Collaboration, there we go, collaboration. In 2013, they reported making the first solar cell that gives off extra electrons from high energy visible light, which makes up almost half of the sun's electromagnetic radiation at the Earth's surface. According to their estimates, applying their technology as an inexpensive coating on silicon solar cells could increase efficiency by as much as 25%. They say understanding the mechanism at work would enable them and others to do even better. Excitation fission has now been observed in a variety of materials. We can't rationally, rationally? That's not the right word. I am mispronouncing that particular word. We can't rationally design materials and devices that take advantage of excitation fusion until we understand the fundamental mechanism at work, which they now have discovered. To support his theoretical study of the electronic behavior within these PVs, they used data gathered from the other team. The samples were made up of 4 types of excitation fusion molecules, decorated with various sorts of spinach. Come on, guys. Bulky side groups of atoms that change in the molecular spacing without altering the physics or chemistry. Actually, that's a good deal. They've used this one word, spinach, to describe this process. To detect fusion rates, which are measured in femtoseconds, 10 to the minus 15 power seconds, the MIT turned to another group of people. This is Professor Lester Wolfe at the Cavendish Laboratory of Cambridge. Okay, let's back up. The first guy, Mogia Bawandi, the Lester Wolfe Professor of Chemistry and Special Equipment at Brookhaven National Lab and the Cavendish Laboratory of Cambridge University under the direction of Richard Ford. Anything? Let's see. The researchers are pleased to announce the agreement between their experimental and theoretical data, especially given the systems being modeled. Each molecule has about 50 atoms, and each atom has 6 to 10 electrons. These are complicated systems to calculate. That's the reason 50 years ago we couldn't calculate them. Trying to look for anything. That's pretty much it. So there you go. Hopefully a way of increasing solar power efficiency by 25%. Anything anybody else wants to talk about? Yeah. All right. Then we will end this recorded Hangout. And of course, everybody will be able to view it later on our channel, or on our YouTube channel. Once again, I learned a lot today, particularly when Dale started talking about how this mysterious DNA works. You missed that, AJ, didn't you?
Yeah, I might have to revisit that.
Yes, there are this amazing, we jump to the story. I'm not going to go over it again, but scientists creating a new bacterium, a completely new kind of bacterium, adding letters to DNA.
Uh-oh, yeah. I'll have to read that. Well, I enjoyed it.
Okay. Well, the This Week in Science podcast, Hangout, will hopefully be happening fairly soon. And enjoyed the Hangout. Had too many stories to go through this past week. There's a whole bunch we never even got to.
เราชมที่ก่อนสิเราชมที่
Okay. So anyway, hopefully see you guys next week and ending the broadcast.